Hi y'all, I'm gonna hope that y'all can hear me very clearly. If not, just let me know. Anyways, my name is Shiloh Warthen and I'm a senior at Montrose High School. This summer I had the opportunity to study in the immunology department at Scripps Research Institute, more specifically in the Briney Lab, which we'll get into. So the Briney Lab operates as a subsection, of the larger Burton Laboratory, which you're gonna hear about from Azari, my amazing lab mate, love her. And it basically just focuses on analyzing human immune responses to infectious diseases. That's going to cover HIV, SARS-CoV-2, which causes COVID-19, Lassa virus, and malaria. And they use this to develop antibodies that specifically target these pathogens. It's a process called reverse vaccinology, and I won't get too deep into it because we will be here all night. So what does this mean for my internship? I did a lot of things, including learning the ins and outs of the processes of the central dogma, which is basically how you just take a little bit of DNA and turn it into a lot of antibody proteins. And then I also did some work with some AI models called AlphaFold and protein MPNN to do some structure analysis on proteins, as well as in vivo protection assays, which is going to be mouse work and rat work and all that. Azari will get more into that. And then I also did a lot of protein and cellular characterization on those antibodies that we did cultivate. That's going to be things like ELISAs, running gels, all that fun stuff. So to get into a little bit of the stuff that separates my experience from my lab mates, that's going to be in silico vaccine design, which basically just means it's on the computer, it's done with AI models. So we use computer modeling to predict the structure of proteins. AlphaFold takes amino acid sequences and turns it into structures. Protein MPNN takes those structures in turn and turns them into amino acid sequences. We basically just run these proteins through it many times until you get a lot of different amino acid sequences from antibodies that interact with HIV envelope trimers, which is the virus we're looking at, and ones that don't. You can compare the amino acid sequences of both in a process known as sequence logoing, and this allows you to see which parts of the amino acid sequence are actually folding into the important binding sites of the protein that are actually going to neutralize our virus. So yippee. And then life in San Diego, I was one of 5. It was very fun. And so this is going to be a lot of the lab work we did. I'm pretty sure those are proteins. Don't quote me on that. And then a lot of ELISAs and whatnot. Outside of the lab, we also did a lot of fun things. You've already heard some of them mentioned. We went to the beach a lot. We went to lots of farmer markets. Azari took us to the weekend concert. Also, shout out to Lana for letting us use her grandma's apartment to stay the weekend in LA. That was super nice and made it a lot easier. I also had the opportunity to do some stuff outside of the actual lab with lab members. So me and Azari did try to learn to surf. Our results were a little bit mixed. I'm not a very talented surfer, unfortunately. Maybe in another life, that could have been me. But as far as things that surprised me in the lab, there's actually a lot of flexibility in science. Things go wrong a lot more than you would expect them to, and that's okay. People in the scientific world are a lot more okay with that than you would think, and they're pretty used to having to do things multiple times to get them to work. And then my advice for future interns is that it's okay to not know and to make mistakes. I made many in the first few weeks of my lab. I'm pretty sure some of y'all are watching from California. I'm so sorry. Thank you for not kicking me out. I really appreciate it. And then take initiative and ask questions, and believe in yourself. A lot of these things have been said over and over again by interns, and you're going to hear them probably plenty more times throughout the night, but they really are important. You get out of this what you put in, so put in everything you have. And don't take yourself too seriously. You are probably like 17, 18, you're a high schooler. They expect you to be there to learn, and so they don't expect you to know everything. And you're also there to like have fun and find an interest and a passion in the field of STEM. So go for that. And then, yeah, I kind of zoomed through that, but a big thanks to everyone I worked with, including mentors from both the Burton and the Briney Lab, and then the Pinhead Institute, including Sarah and my girls. They made it so fun. Love y'all.